me a second to get there, guys. And we're already at 9K likes. Thank you guys so much. Will you be teaching chem? I will be teaching chem, and also another co-lead tutor will be teaching chem. So we'll be doing it together. But I think for most of chem, I'll be teaching it. We do have a crash course for chem, so if you guys want to join. Okay, this is the May 2023 Time Zone 2 HL paper. So I'm not sure if this was the one for my exam, but it was either time zone 1 or time zone 2. I don't remember what time zone I was. Let's go ahead and get started. It says, what cell component is found in eukaryotic cells but not prokaryotic cells? Let's see what you guys think. I'm going to give you a couple of seconds. And I'm sorry if I missed your comment. I was just busy trying to find the past paper. But it's good this way because then we're able to do what you guys want to do. Okay, a lot of people are saying A. Nice. What is making you say A? So try to change the way you answer things. So don't just think, okay, the answer is A, I'm going to move on. Especially when you're studying from now on, since the exams are so soon, say A because it's this. And I want you to tell me why it's A in the comments. Or if you think it's B, why is it B? This is biology. Yes, it is. Maybe if someone can write M23 HL Time Zone 2 past paper, I can pin it up. And if you're SL, you should also be able to solve this. So if you're an SL student who stayed along, you should be able to solve this. Okay. Someone said it's B, prokaryotes don't have mitochondria. Exactly, very good. Why do we know that prokaryotes don't have mitochondria? What is the special characteristic about mitochondria that allows us to make this connection? So, very good, guys. Prokaryotic cells do not have mitochondria because they're what? We can say they have they're membrane-bound organelles. Perfect. If you mention membrane-bound in your in the comments, or if you've even practiced saying it in your head without typing it, this is exactly the type of thinking you should have when you're answering these questions. So you guys are correct. The answer is A. Good job. Now, do prokaryotic cells have DNA? Yes, they do. Okay, this one just has a nucleus. This does not have. They have like linear versus circular DNA. They have different types of DNA, but they do have DNA. So careful with that. Ribosomes for protein synthesis, they both have ribosomes. But prokaryotes have which ribosomes? What about eukaryotes? Tell me and do some active recall in the comments. 7TS, very good. 7TS, the smaller ones are for the prokaryotes. The eukaryotes have ATS, very good, guys. And then the cell wall to maintain shape. That is correct that it's in prokaryotes, but it's also in plant cells. So it's in both of them. That's why our answer is A. Okay, we're going to move on. This is in your, it's not so much in your new syllabus because you don't need to know by name that there's a Singer-Nicholson model and a Davidson-Danieli model. You don't necessarily need to know it for the new syllabus. But they did want you to know about the history of membrane proteins. So we're going to do this question, but we're not going to put so much emphasis on the different models, just about, actually, I don't know if I should skip this question. I might skip it just because you don't need to know these by name. And there's no need to stress you about something you don't need to know. Okay, let's do this one. It says, the diagram shows protein channels involved in the passive movement of a substance into the cell across the cell membrane. What describes this movement? So, what am I looking for? I'm looking for passive, and I'm going to analyze this diagram. What are these called, guys? This is biology. Is this A-level? No, it's IB. Someone said D. Good. They are protein channels. So these are channels. They're not pumps. Okay. And we know it's passive. So we know that there's not going to be any ATP used. So A says energy of ATP is used to transport substance in the cell. No, that's not correct because we know we don't have ATP use. Okay. Substances can move from areas of low to areas of high concentration. Is that correct, guys? For passive movement, is it from areas of low to high, or is it high to low? No, exactly. Good. So we can cross out B. Proteins ensure that the movement of substances is only in one direction. Do these ensure that? Or is it movement in both directions? Let's say you didn't know, because this is not something you usually talk about. Good. If you've answered no, they don't. They do both directions. Very good. You got the answer correct. But let's say you didn't know. Let's have a look at the option D and see which one sounds better. This one says net movement occurs until the concentration in and out of the cell are equal. Is that true? Is it until net movements are, net concentrations are equal? Or is it what? Someone said yes. Good. And why are you so confident in saying yes? Why are we so confident in saying that the concentrations are equal? I'll give you guys a hint. Whenever you see something that says net movement occurs until concentrations are equal, that is true. The movement continues, the overall movement continues until those concentrations are equal. This is only correct because they've used what? They've used net movements. Okay? If they just said movement occurs until concentrations in and out of the cell are equal and then it's just one side, this would be incorrect because then it would mean that it's majority just like happening in one direction. But that's not the case, okay? They're actually happening in both directions, and we do these directions until we see a net movement of the concentration of the cells when they're equal, okay? So it's areas of high, you move from areas of high concentration to areas of low concentration, and the movement can occur in both directions. We know that with channel proteins, they can occur in both directions, so that's why C is incorrect. And then here, the reason this statement is correct is because we have this word net. So net means overall. So basically, they're just saying that for example, two of these pentagons are moving this way, and two of these pentagons are moving this way, but one of them is moving backwards. But the net movement is that more of them are moving inside the cell. So a higher concentration of the one we want to move inside the cell is being able to move inside the cell. So when you say net, you're basically not limiting it to the idea that the proteins, or sorry, the ions are moving in one way, because that's a misconception. It's a very common misconception that IB love to bring up in questions, and this is a good question to practice for, because we see that. Okay? Does that make sense, everyone? Thank you for the rose. Thank you so much for the rose, and thank you guys for 11K likes. Please let me know in the comments if that makes sense. If that doesn't, this is HL, but if you're SL, you should be able to solve this. This is biology HL. Yes, it does. It does make sense. Okay, perfect. So watch out for the word net, and then you should be good for future IB questions. Okay, what is evidence for endosymbiotic theory? This is a very, very important theory. This is a very common one. So a lot of you guys always ask me, what is a common IB theme? What's a common IB subject that keeps coming up every single time in exams? Endosymbiotic theory is a really good one. And if it's not directly asking you, what is evidence for endosymbiotic theory, they ask you, what does this diagram show? And it shows like a prokaryotic cell ingesting another prokaryotic cell. And like you can see that the prokaryotic cell is engulfing a chloroplast, for example. Well, prokaryote that is photosynthetic. And then it becomes a chloroplast. Okay, a lot of you guys are saying A. Good. Now tell me why it's A. Prokaryotes evolved before eukaryotes. Is that an evidence? I mean, that's a fact, but it's not really evidence to tell us that eukaryotes came from prokaryotes. There's no reason to say that this connects at all. Unicellular organisms exist both as prokaryotes and eukaryotes. Okay, that's true, but again, that doesn't tell us anything. Prokaryotic cells have no double membranes. Okay, that's also true, but that's not really evidence. We know the evidence is that mitochondria contains its own DNA. Because we had one prokaryotic cell, okay, that engulfed another prokaryotic cell, and this is our mitochondria. And because this one engulfed this one, this mitochondria, or this future mitochondria, still maintained its DNA, and that's how we know that endosymbiotic theory could be true. Okay, nice. All right, I'm going to move on. Someone said this is easy. Good. It's the fourth question, so I'm happy this is easy for you guys. The image shows a cell from a root tip of an onion to N16 during late prophase of mitosis. How many chromatids are present in the cell? This is a hard one. But if you know your definitions, you should be able to answer this one. So this is all about the definitions. They're trying to trick you with this one, so make sure you don't get tricked. Keep an eye out. Thank you guys for 12K likes. What is endosymbiotic? So endosymbiotic theory is this theory that basically says eukaryotic cells evolved from prokaryotic cells who engulfed other prokaryotic cells and became a eukaryotic cell. Okay. What are we thinking? Someone said 32. This is not GCSEs. This is IB biology. It's biology HL paper. Okay, why is it 32? What is specific about this one that it's nice, guys. You guys are all saying 32C. Perfect. Okay. So basically, the way I would answer this is we have a chromosome here, and this chromosome is made up of two chromatids. These are usually called sister chromatids, sister chromatid pairs. So both of them together are called sister chromatids. One of them, however, is called a chromatid. Okay, so two of them attached together as sister chromatid. One of them is a chromatid. So if it's telling me that the diploid of this mitotic cell is 16, that means there's 16 chromosomes, 1, 2, 3, 4, 5, whatever, so on and so on and so on and so on, and each of them have two chromatids. So 16 times 2 gives us 32, and that's why C is the answer. Did that make sense, everyone? So someone said it's because DNA gets replicated, so 16 times 2, that's not really it. It's because of the chromatid, so careful. It's not because it replicates. It's because there's 16 here, but there's one chromatid for every pair. Exactly. It says 2 and you always have to do 10. Exactly, because it's telling you we're in late prophase. Okay, nice. Moving on, it says which drawing represents beta D glucose? So yesterday we talked about the difference between alpha glucose and beta glucose. 2N means it's diploid. So it means we have, so for example, for humans, a haploid is 23. So a sperm cell has 23 chromosomes and an egg cell have 23 chromosomes. And they join together that when they fertilize, they create 46 chromosomes, and that's known as the diploid chromosome number. A lot of people are saying A. Okay. Why is it not B, guys? Why is it not B or D? What's wrong with B and D? Tell me. Someone said it's a ribose, exactly. So it has 5 carbons, exactly. It has 5 carbons. So 1, 2, 3, 4, 5. And we know that's incorrect. And this one's incorrect. So the first thing I would have done is definitely crossed out this. And then I would have looked at the difference between this one and this one. And I know my beta glucose, the hydroxyl group on carbon 1, is facing upwards. So that's why my answer is A. Good. Nice, guys. Next, ooh, this is a good one. And actually, they've highlighted in the new syllabus that students must know the definitions. You must know the definition of a proteome, a genome, and then there's a third one. Can anyone tell me the third definition you have to know according to the new syllabus? If anyone's from the crash course, hopefully you guys get this right. Okay, but going back to the question, it says, what is the proteome of an individual? Very good, guys. A transcriptome. Exactly. So these are 3 ones that they've highlighted in the syllabus. In the old syllabus, no teacher ever taught their student what a proteome is. We always just skipped to like genome because it wasn't so highlighted in the syllabus. And IB recognized that. So they always brought up proteome, or they tried to bring up these words in the exams because they knew students would struggle with this. So back then, this was a really difficult question to get just because you were like, what the hell is a proteome? I've never heard this word before. But now in the new syllabus, it's really, really highlighted, and you need to be able to compare and contrast those 3 definitions. So for example, the way we taught it to our students in the crash courses, we made like a table with each word definition and the context in which you have to compare them. So in your studying, I would really recommend for you guys to put proteome, genome, transcriptome in a nice table and compare them together, because that's a potential way that IB can ask you according to the new syllabus. Okay, what are we thinking for this one? Where do you get these mock exams? I just get them online. It's like a past paper website. If you guys have past paper websites, you can share in the chat. That would also be helpful. Okay, C. So a proteome, this is how I answered it in my exam because I didn't remember what a sorry guys, I didn't remember what a proteome was, and I was like never heard it because my teacher never taught it to me, and it wasn't highlighted in the old syllabus. I remember knowing what a genome is, but let's say you've never heard about this in your exam. How do I know what this is? Well, first it has the word prot. So I know it's going to be something with proteins. Okay? So the best guess you can make is, if you don't know the answer, is try to find the answer that is just focused around proteins. So this is a good way to teach you guys another method of multiple choice. Let's have a read of the answer. It says the amino acids unique to an individual making up the proteins in the cell. But the key thing here is the amino acids. Okay, so we're a bit iffy about this. The way in which an individual's peptides are folded into three-dimensional structure. Okay, so we're talking about polypeptides. The protein synthesizes an expression of an individual's genes. Okay, all possible combination of amino acids an individual contains. So in all of these answer options, the one which highlights proteins is answer option C, and that's actually the correct one. So you could have answered it like this by guessing, or you could have just remembered that the proteome is basically all the proteins an individual synthesizes. Okay, that's why we know our answer is C. Can anyone tell me what a genome is in the chat? Do some active recall. So if a proteome is all the proteins an individual synthesizes, a genome is what? Is the whole genetic information. Okay, now can anyone tell me what a transcriptome is? All the possible genes. Very good, guys. Can you guys guess then what a transcriptome is? All the mRNA. Perfect, exactly. Is all the DNA you're actually transcribing. So transcription is the process of creating the mRNA. So you can kind of make this connection because it's a transcriptome. All right, good. Moving on, we're going to do this question. This is a really good question to get also on the exam because I would say it's one of the easier topics, which is enzymes, but they're trying to complicate it with the graph. But we have to remember that we know this topic well. It's one of the easier topics. Okay, it says in the grass plant, and that's the name, the enzyme amylase breaks bonds in polysaccharides during germination. The graph shows how the activity of the enzyme varies with the concentration of polysaccharide. If you're SL, you should also be able to solve this. And it says, what is the reason for the curve leveling off like this? What is the reason? We have a C, we have a D. This is the bio HL paper. Okay, nice. Someone gave the explanation. D enzymes are the limiting factor. D. Okay, good, guys. A bunch of you guys said D in the comments, which is perfect. So it is all the enzymes are active sites are occupied by substrate. Okay, let's have a look at this. So here I have my enzyme 1, enzyme 2, enzyme 3, enzyme 4, and enzyme 5. Okay, and I'm monitoring the rate of reaction with the concentration of polysaccharides. So concentration of polysaccharides on the x-axis, the rate of reaction on the y-axis. So this is my 5 group of enzymes. I'm going to add concentration of polysaccharide, which I know is the substrate because it's telling me amylase breaks this polysaccharide. If anyone is here from the crash course, do you guys remember what ASE stands for? Or maybe you've learned this in class. Usually whenever you see ASE, what does this mean? An enzyme, good. So I know that this is my enzyme, because sometimes they won't mention it's an enzyme. You do have to know that ASE means it's an enzyme, exactly, good. So this is our enzyme, and our substrate is our polysaccharide. Okay, so I've added my polysaccharides, and my rate of reaction is increasing. I'm going to add more polysaccharides. Okay, my rate of reaction is increasing. I'm going to add more polysaccharides, more, okay? And each one of them goes to an enzyme. But I get to a point where I keep adding more and more and more. But what do we see happening? We see that there's not enough enzymes for us to keep like attaching to the active site. So How do we say that? We say that the active sites are saturated. That's a keyword that you have to use. You have to know because IB uses a lot. Saturation. When we say the active sites are saturated, it means that all the active sites are being in use. And that's why D is the correct answer because that's what it's saying. Does that make sense, everyone? Will this be on YouTube? It will be on YouTube. Okay. Please let me know if that made sense. I'm going to move on. And we're going to do this one. Yes, it makes sense? Okay, good. The anticodons for 3 tRNAs and the amino acids they carry are shown in the table. Which base sequence of the mRNA molecule would code for arginine, leucine, valine, and tripeptide? So arginine, leucine, and then valine. Okay, that's what we're looking at. And we're looking at the mRNA molecule. Base sequence of mRNA. And they've given us the anticodon. So basically we're trying to find the codon. Okay, people are saying B. This is Bio HL. Thank you guys for 13K likes. Remember, our goal is 25K. We can try to reach that. If we can't, that's okay as well. D? Okay, a lot of you guys are saying D. Perfect. Okay, let's have a look. So our tRNA molecule is the thing that carries, what does it carry at the top of it? Do some active recall. Tell me what I'm drawing here. And here's my mRNA molecule. And I know this is known as my anticodon. And the 3 base sequences here are known as my codons. Okay, so if my anticodon has complementary base pairing, I know that this is going to be here G, C, A. I know my mRNA molecule is going to have the exact opposite. So this is going to have C, G, and then of course A would usually bind with thymine, but here remember it's mRNA, so it has to be U. So if you saw this answer, the way I would answer this in my exam is before even thinking about the answer options, is when I've read mRNA molecule, I'm going to already cross out anything that has T. Get that out of the way. You've already increased your chances of getting the answer correct because then you just have 50% chance of getting the answer correct. And then now I know my answer has to be D because that is exactly what's complementary to my anticodons. If you did say amino acid, you are correct. Good job. That is what the tRNA molecule carries. Okay, this is part of the new syllabus. So hopefully you guys, this is also new syllabus. So hopefully you guys are able to answer this one. And this is also SL content. So all the SL students should be able to solve this as well. Which graph represents the action spectrum for green plant receiving only blue light? Okay, we have an A in the comments, a B, an A. Okay, tell me why you think it's A. So remember, in your... Okay, I think people are saying A because they're used to seeing the blue light and the red light. You're usually used to seeing the whole action spectrum, but you guys aren't used to seeing only blue light. So if your answer is A, try to reflect again on your answer because it's not A. But I see why you guys are getting this confused because we know the action spectrum, the colors are absorbed are in the blue region and in the red region, right? And the green region is the one that's in this section here. Okay, we know this section stands for the blue and this section stands for the red. Okay, so we know only blue light of the action spectrum must be only this section here. So that's why my answer is B. So if you said B, good job. Thank you, Tay, for the rose. Thank you so much. Okay, does that make sense? So usually you're supposed to memorize and be able to actually draw. Now in the new syllabus, they want you to draw this from scratch. In the old syllabus, it was just identify. D is like, you're not supposed to know this. This is, they're just trying to trick you. Like you should not even know what this spectrum is. Okay, so you should be able to identify the action spectrum. It shows you how green light is reflected, and that's why plants are green. We see here the wavelength that is absorbed. So in the blue region and in the red region, you should be able to draw this from memory. Okay. Now, this section is the green section, and you see this dip is, you should know that's the green region, and you should know that's why plants are actually green. Because they reflect green light. Okay? So if this is the spectrum you have to memorize, and usually you know the spectrum shows all range of colors, you have to be careful if they're asking for only blue light, you're only looking at the blue section. Okay, and how would you know that this is the blue section? Well, because you should also know the electromagnetic spectrum. You should know that a higher wavelength means lower energy. And we know red light that has a higher wavelength has lower energy than blue light. So you would know that this is blue, this section is red, and the green is the one that's being reflected. Okay? That's why our answer is B. Exactly, because plants are green, they reflect green light. So how can this show up on your exam? They can show you the absorption spectrum and ask you why plants are the color they are, and you would say like because green light is reflected. They can ask you to draw it from memory, and you should be able to draw from memory. They can ask you in a multiple choice just like this. You get it? Okay, nice. Nice, guys. Okay, perfect. We're going to move on to the next one. It says, which change causes sickle cell anemia? This is also a very common question. You should know the exact change of the base and what change of amino acid it could cause. Okay, I'm getting C's. Someone said D, okay. This is the 2023 paper. We voted in the beginning and people said they wanted to do the 2023, so we're doing the 2023. It's Bio HL. If you want like more say of which paper we should do, come join the beginning. We did like a Wheel of Fortune and we picked this one. Okay, so a lot of people are putting mixed answers A, C. I will be uploading this on YouTube. Okay, so if you said C, you are correct. Good job, Tay. Exactly, C. Very good use of C. Nice. Have you done the specimen papers for this year's syllabus? I have. So you should know the change exactly in an amino acid. Can anyone tell me that change? Oh, yay, someone said they just joined the crash course. Yay, I can't wait to see you this weekend. Very good. From GAG to GTG. Nice, exactly. So you have your thymine replacing adenine in DNA, exactly, and that codes for a different amino acid, and that's what actually causes sickle cell anemia.